Hello, my name is Alexandria Baker. I am a senior at Langston University, and today I will be presenting you about acetylcholine signaling pathway via nicotine. Introduction: Nicotine. Have you heard of Juul pods, vape pens, and cigarettes? All these products contain nicotine and are commonly used today. Cigarette smoking is the most common method of using tobacco. However, many individuals also use smokeless tobacco products, such as snuff chewing tobacco. These products also contain nicotine. The biggest question of all is, why does one use tobacco? Many users have different reasons, but the main reason is for stress relief. Upon research, I found that other people's reasons were because they started at a young age and they wanted to rebel against their parents, or they had peer pressure from friends when they were at a young age and started at a young age and always had an addiction since then. Nicotine is an alkaloid commonly found in tobacco plants that causes addiction and affects the human body in a matter of seconds. Nicotine imitates the action of a natural neurotransmitter called acetylcholine and binds to a particular type of acetylcholine receptor known as nicotinic receptor. This PowerPoint will discuss the cell-to-cell communication, also known as cell signaling, in the human body via nicotine. How does nicotine deliver its effect? To begin, nicotine is typically inserted into the body in two ways. One is smoke users and the other is smokeless users. For smoke users, when tobacco is smoked, for smoke users, it's when tobacco is smoked. Nicotine quickly reaches peak levels in the bloodstream and enters the brain. For smokeless users, it's when the nicotine is absorbed through the mucous membranes in the mouth, taking more time for nicotine to reach peak blood and brain levels. Nicotine is distributed quickly through the bloodstream and can potentially cross the blood-brain barrier to enter the central nervous system, also known as CNS. Nicotine binds to two main types of nicotinic acetylcholine receptors known as ganglion type and CNS type. The ganglion type is a group of neuron cell bodies in the periphery, and the CNS type, as discussed earlier, CNS is short for central nervous system and is the part of the nervous system consisting primarily of the brain and spinal cord. In the next slide, I will discuss to you the ganglion type cell signaling pathway. Ganglion type cell signaling. Nicotine begins by binding first the chromaffin cells in the adrenal medulla. Nicotine begins by binding to the ganglion type nicotinic acetylcholine receptor. This receptor is composed of alpha 3 and beta 4 subunits. These subunits co-assemble to form functional receptors. Nicotine binding to this receptor causes cell depolarization and an influx of calcium through voltage-dependent calcium channels. Calcium signals the release of epinephrine from the chromaffin vesicles into the bloodstream. And as you can see, as you can see in the picture on the pathway diagram, if you start with acetylcholine, you can see that acetylcholine signaling via nicotinic acetylcholine receptors. You can see the different pathways that it can take. And the ganglion type pathway, as discussed, is you start at acetylcholine, you go up, and you see that the receptor is binding, that the nicotine is binding to the ganglion type nicotinic acetylcholine receptor in the adrenal medulla, and from there you can see that epinephrine is released, and then epinephrine is released into the bloodstream. And also on this diagram, you can see that the acetylcholine signaling, the nicotine signaling pathway. You can see the central nervous system is at the bottom, and you can see like the different effects or what it affects all. And that will be discussed in the next slide. Now, as you may notice when first looking at this PowerPoint, you see the picture. As you can see, the CNS type is more complicated and longer than the ganglion type. So to begin, dopaminergic neurons in the CNS, nicotine binds to the CNS type nicotinic acetylcholine receptors. The CNS receptor is composed of alpha 4 and beta 2 subunits. Alpha 4 and beta 2 is a nicotinic acetylcholine receptor implicated in learning. By nicotine binding to CNS type nicotinic acetylcholine receptors, depolarization and the release of dopamine from the cell through the snare complex takes place. Snare's complex primary role is to mediate vesicle fusion. On dopaminergic terminals, dopamine then binds to dopamine receptors, and GI alpha is activated, causing a feedback loop to obstruct release. GI alphas are classified as G protein coupled receptors, but it is inhibitory. It acts to inhibit adenyl cyclase with a subsequent decrease in intracellular levels of CAMP and a reduction in activity of PKA. So I don't have a mouse to point at, but to first begin, I want to show you that the process right now that they are talking about. And as you can see, starting at the very top, you see nicotine. And you see that nicotine is binded to, or nicotine goes to the CNS receptor, which is composed of alpha 4 and beta 2. On there, it's labeled CHRNA4 and CHRNB2. And as you can see, that's on my left-hand side. And that causes depolarization. From the depolarization, it goes to the snare complex. From the snare complex, you can see that dopamine is released. Sorry, kind of got ahead of myself on that one. But you could see the depolarization and the release of dopamine from the cell through the snare complex. CNS cell signaling continued. PPP1R1B, also known as dopamine and CAMP-regulated neuronal phosphoprotein, or DARPP32. PPP1R1B can inhibit two things. One is serine threonine kinase, which is abbreviated as PPKACA or PKA, or serine threonine phosphatase, abbreviated as PPP1CA or protein phosphatase 1. And PKA is a protein kinase that has several functions in the cell, including regulation of glycogen, sugar, and lipid metabolism. And threonine is an amino acid. By PKA at threonine 34, PPP is 1R1B is phosphorylated. Which in turn inhibits dopamine secretion through the snare complex. There is another way that this can happen, and as I put on my slide, I demonstrated the first way, and the second way that this can happen is when PPP1R1B is phosphorylated by CDK5 at threonine 75, it is converted to an inhibitor of PPKACA. The binding of dopamine to D2-like dopamine receptors leads to inhibition of adenyl cyclase via G protein and GNAS GAI1 and decreases PKA stimulated phosphorylation of DARPP32 at threonine 34. This in turn relieves the inhibition of protein phosphatase 1 and inhibits further dopamine release. So I'm going to go back to the picture and explain to you what the PPP1R1B signal transduction goes. So from the release of dopamine in from the dopaminergic neuron, you can see the release of dopamine. The release of dopamine then binds to as you can see, dopamine binds to dopamine receptors and on dopaminergic terminals and activates the G1 alpha or the GI alpha, which is labeled as GNAI1 in the pathway graph. From there, you can see that the alpha inhibits adenyl cyclase, and that inhibition leads to another process of the threonine of the phosphorylation of PPP1RB is phosphorylated by PPPKACA at threonine 34, which is in the middle, and you can see the P in the phosphorylation, you can see the THR75, which this inhibits the dopamine secretion through the snare complex. As you can see, the PP starting in the if you start in the very middle of the triple P1R1B, you can see that it can go to two options. As I stated earlier, it can go to the threonine 75 phosphatase or the threonine 34. And you can see that there is in the threonine 34, there's an inhibition in the snare complex, which is the red lines. And then in threonine 75, you can see that it is an inhibitor of PPPKACA. This is not going. Okay, now we're down to our conclusion. The intake of nicotine in the human body quickly causes peak levels in the bloodstream and can cross the blood-brain barrier. Nicotine mimics the mechanisms of a natural neurotransmitter known as acetylcholine. Nicotine binds to two main types of nicotinic acetylcholine receptors known as ganglion type and CNS type. So, in conclusion, I put a simplified version of the cell signaling processes that ganglion type and CNS type goes through. And I put the results of what ganglion type along with CNS type could present. The ganglion type begins in the chromaffin cells in the adrenal medulla. Nicotine binds to the ganglion type nicotinic acetylcholine receptor. Voltage-dependent calcium channels are open, causing an influx of calcium and depolarization. Calcium initiates the release of epinephrine from the chromaffin vesicles into the bloodstream, leading to an increased heart rate, blood pressure, and elevation of blood glucose level. The CNS type dopaminergic neurons starts with dopaminergic neurons in the CNS. Nicotine binds to the CNS type nicotinic acetylcholine receptors. The binding of the receptor causes cell depolarization and a release of dopamine from the cell through the snare complex, which results in the release of dopamine causes a pleasure response, resulting in nicotine causing and addiction. Lastly of all, I want to leave off with, overall, nicotine is a constituent of tobacco smoke that exerts its psychoactive effects via binding to nicotinic acetylcholine receptors in the brain.